Many things are really exciting about microbiome research, but I think the most exciting part is that our knowledge and understanding of the gut microbiota has improved so much over the past few years, I would say. It's been more than 50 years since we learned how to grow the bacteria from the human gut, and our understanding only came from being able to culture in the laboratory. And since the 1970s and early 80s, we've gone from culturing to looking at the genome, the genes, and extracting genes from gut bacteria, and our knowledge improved quite significantly. We were using one marker gene and analyzing and getting to recognize the bacteria that are really present, and not only those that we can culture that represent approximately 20%. And today we can extract the whole combined genomes of the gut community and sequence it and analyze its functionalities. And so I think this is in a way, this is a revolution of the way we can look at the microbiota, and this is really marked. So my own focus in research is the functionality that are expressed by gut bacteria and how they can act on human health by their interaction with food, by their interaction with other microbes, and by their interaction with human cells. And metagenomics, our ability to work with the genome fragments from gut bacteria, is again opening a completely new perspective for this research. We can now, for example, address questions on the mechanism of interaction between bacteria and human cells in the gut, or also with tissues of the human organism that we could never address before. We can explore the signaling that happens or occurs between gut bacteria and epithelial cells, for example, and try to identify the molecules of the crosstalk between bacteria, our bacteria, and our cells and our genes, basically. Well, I would say that there are two main domains of perspective. One is health nutrition. So the improvement in knowledge that we're gaining at the moment will allow to improve our ability to rationalize strategies to manipulate the microbiota for human health via nutrition. And this means that as we identify mechanisms of interaction between bacteria and the human organism, we know exactly where we want to drive this crosstalk between microbes and human cells, and so we can have strategies to rationalize this. And the other aspect is human health, more classically as we see it with the pharmaceutical industry, in that understanding how, for example, gut bacteria talk to human cell allows to pinpoint targets for new drugs, new therapeutics, basically. And if we know how a set of gut bacteria can modulate the immune function, the defense mechanisms of the human organism, then we can actually apply those new knowledge to develop new drugs. So this is the second area of improvement that I can foresee.